So I'm very happy to introduce which is one of the topics where some of our students are studying their program also. Actually, from Mysore. So that is how we are got in Mysore. She studied here and later she got a one in Kolkata. To just give a brief for the way she has done her BSc program in Science College and later she did her MSc in Environmental Science in Mysore. She is the first graduate of the MSc in Analytical Science in Mysore. Later on, she did her PhD program on Science for Science, and then she moved to Boston for her postdoctoral work. She came back to India, and then she got an appointment in Kolkata at the faculty and now she is a professor, and her area of interest is whales and terrestrial animals. So probably we also, when we were young, would have got a very good curiosity liking and they move in the direction and together. And so many things. They do teach us so many things in our life also. So it is very deep background of us. Actually, for documentation, they work with a lot of and very good experiment of publication impact. And so they mentioned about the award numbers. She has got emerging national award from England Society and International Equality Conference in 2011. And then UCL Research Fellowship and Executive for Research Award from CSIR and UGC. And she is a first time holder of the for her MSc program. Her selected application is like an estimate for a very design. So coming to the selected applications of one of the applications is titled as High Made Understood Colony Resilience to the loss of tangibility during relocation in an alien land. It was published in Royal Society Open Science Journal. Then she has got dual response to next studies during monsoon near an Indian land, published in scientific report. There's another publication that I can mention over here. The title is Response to a Change in the Target Net during an relocation, published in Journal of Experimental Biology. So you can just understand the quality. It is I invite myself Sumana Annagiri.
Thank you very much. And do you think I need the mic or can I just? Can I? If you cannot hear me, just let me know. Raise your hand or something. Otherwise, I can just go ahead. Okay. Thank you, madam, for the kind introduction. And it is a great pleasure to come here to Yuvaraja College. Like you heard, I grew up in Maharani's, which is right next door. So it brings back a lot of fond memories. They are just memories at this point in time because I finished in Maharani's in 1993. So a very long time back. But it's lovely to be back and the rooms look very similar. So it does bring back a lot of memories. Also, it's a privilege to be talking in front of so many eminent teachers and my own teachers as well. You feel like it's a full circle, you're coming back to something that you started off with. So I'm very happy to do this. whatever resources and information that we lacked at our time, it was well compensated by the dedication and effort of our teachers. And without those level of dedication that the teachers showed us, there was no way that any of us could prosper and go on to become international scientists. That could not have happened given the resources that we had. But despite all of that, as Madam has told you, I was the first batch of bioscience MSc. And you can imagine what was the states of resource, hostel, everything was a big question mark. But the dedication of our teachers and their enthusiasm is what motivated us to do science and carry on with our lives. So I feel particularly privileged to acknowledge what these teachers have done for us, and hopefully in turn we will also be able to bring back some 5% of that enthusiasm to the system from where we emerged. So thank you for the invitation. Now, in today's talk, I want to introduce you to ants. In fact, ants do not need introduction. I'm sure everybody in this room have seen ants and been pestered by ants, and at one time or the other tried to get rid of ants also. Correct? So all of us have done this. But I want to tell you what is special about ants and how my lab is doing research with one particular species of ant and what is it that we have discovered. So I'll try and give you a little glimpse of that. But before going there, I wanted to give a little introduction to IISER Kolkata, the host institute from which I come. Your madam was telling me that some of you have gone there, or some of your seniors have gone there for a summer program or for an internship, right? Some of you, but at least I'm sure all of you here have heard the word of IISER at least. Yes. So IISER is a relatively new thing. It started in 2006. Unlike your esteemed college, which is 1927, I understand. So we are just a recent origin, 2006. And IISER Kolkata and IISER Pune are the first two IISERs to get started. But now we have several others, about 7 of them. And the thought process was that IISERs will be doing fundamental sciences and will be a parallel to people who are inclined in a scholastic manner as compared to engineers and IITs. So this is a parallel system, like the IIT says for engineering, IISER says for sciences. That is what the vision was. And we try and continue to do that. But I want to give you a little introduction to IISER Kolkata specifically. So IISER Kolkata's journey started in 2006, and our basic motto is to have integrating different disciplines of research, disciplines like chemistry, biology, physics, earth science. These are all combined. First year, everybody has to study all of these subjects. And even later on, the students choose what courses they want to do, and then we make the timetable. Right? That is what we mean by interdisciplinary. So if somebody is a math major and she wants to do a course in biology, then that person will get the chance to do it, and the timetable is made such that that can be accommodated. So we really mean to be interdisciplinary and not have these divides even within biology or between biology, chemistry, biology, physics, or statistics or maths, earth science. These are all one integrated component of science. So that is one of the things we do. The second thing that we want to integrate is education, right? Textbook learning with hands-on research. So this is another thing that we like to combine. Many BS students will come to our laboratories and do their own projects, and in fact even get a publication when they're going away with their MSc. Because they're so interested in the research that is happening, and they do research hand in hand with education. These are not two separate things, but we can integrate them. That is the principle of IISERs, and that's how we follow it. Just as an understanding, we have BSc, BSMS programs where 12th standard students enter, This is our flagship program. This was something that was lacking in our community before. So a large number of students, almost 300 students enter for BSMS program in every year, and they have to do many national level tests in order to get there. Then we come to integrated PhD program, which is another 7-year program where you do 2 years of masters and then you continue with your PhD within the same department. Okay. Then there is of course PhD program. So if you finished your masters, then we qualify national level exams, and then there are a set of interviews. And if you qualify that, you enter for the PhD program, and that will be approximately 5 years. If you're in biology, it tends to take a little longer. I don't know whether it is the same in the university as well, but in our institute, biology programs take a little longer to grant their PhDs. And then we have a postdoc program as well, where PhD students can come and enter. So 5 different departments: biological sciences, chemical sciences, mathematical sciences, physical sciences, and earth sciences. This is what our gamut is. At present in IISER Kolkata, there are about 200 faculty members, very well qualified, recruited from all of them Indians, but having education across the globe. And we have about 2,500 students, including the BSc, MSc, Masters, and PhD programs and postdoc programs. That is the kind of gamut that we work with. This is our main teaching complex, and this is where most of our activities for teaching occurs. And we have a brand new campus. We are the ones to construct all the buildings there. It's about 200 acres. A small part of it is developed, and the research complex, the residential complex, everything is within this area. And this spot is about 50 kilometers from the center of Kolkata. So it's actually not in Kolkata, it's in the outskirts of Kolkata, about 50 kilometers away. All right, so that was my little introduction to IISER. If any of you want any additional information about it, I will be happy to tell it at the end of the talk. We can have a discussion. Now, if you're ready, we will go back to the title, which I'm saying Running with Ants. And we will be discussing our journey, our lab's journey with ants. The first question that you can ask me is, why am I studying ants? Right? I'm a behavioral biologist by training. There are several organisms. Right? There are wonderful big elephants, right? Fantastic societies they make. Tigers, you name it. I mean, pandas, whatever. There's lovely animals, a lot of animals. And of course, some plants as well, which we can study under the title of behavior, some components of the plant's life. So you can always think, why did somebody choose an ant? It's there everywhere. Even our grandmothers know about ants. Right? They know many stories about ants. So why study ants? Ants form very complex societies. And ants are not the only organisms which make societies. There are several other organisms which make particularly complex societies, which are technically termed as eusocial societies. Okay, so ants, of course, then there are honeybees, there are wasps, and there are termites. I put one image of each of these things if my pointer works. But I'm sure you have seen all of these organisms right within this college itself. You don't even have to go further. Right? I'm sure there are termites here. Agreed? They may not build such beautiful mounds, but they're very much there. Right? Wasps, have you seen wasps around? Certainly. And honeybees? No doubt. Correct? Now, anybody here are the students who have been bitten by any honeybee or wasp? Yes? So you've had close encounters with these societies. They are very much a part of where we live. Right? It's there. Now, these societies are really very, very well organized. What do I mean by well organized? In all of these societies, the vast majority of individuals are all females. But only one or a very small number of females actually will reproduce. They will mate and reproduce. The rest of them are sterile female workers, right? So, in an average honeybee colony, you will have 35,000 individuals, right? 35,000 individuals. And in all of this, only one, the queen, is reproducing. All the others are sterile workers. Now, there is lots of needs when there are 35,000 individuals in one small location. And the queen is laying eggs. In a day, she lays about 200 eggs at least. And the eggs will develop into larvae, right? And larvae will develop into pupa, correct? Like any other holometabolous insect, they will develop into pupa. The pupa will emerge into adult workers. So, the lot of work for the defense of the colony, for feeding the larva, for taking care of each and every cell, to remove parasites, to bring all the food that is required, to bring the water required, is huge amount of work. So the question is, in order to get all this work done and be successful, honeybees need to be well organized. Right? So is the case, but in smaller numbers for wasps, and in termites also, there's a large number of animals that live in every termite mound, and there's only one queen in the termite mound. So there is division of labor, reproductive division of labor for sure, because only one individual is doing reproduction and others are all workers. All right. So if you're interested in societies, humans also of course form a society, then you can look at other creatures which live in groups and in societies, and the pinnacle of these societies are eusocial societies, like what I've explained over here. Ants belong to this eusocial system of societies. So, another reason why one should be thinking about ants is that ants are present pretty much everywhere in the globe. So, the regions that you see which are marked in different shades of brown and red in this figure will tell you what is the density of ant species in that part of the world. And like you can see, India has got enormous diversity of ants, right? And how many people study it? Very, very few, right? There may be less than one handful of people who specialize in studying ants. And even in that one handful of people, what they will be doing is taxonomy, where you will be labeling what species what belongs to. And this is a very, very important job. If that is not done, then others cannot proceed. So this is the job that most of the people will be doing, even the handful of people who work on ants. So we have great diversity of ants in our country, and pretty much nobody looks at it. And I am interested in understanding just one species of ant, but I want to understand it at a great detail, at a depth, right? And that is what my purpose is. And I'll try and tell you some experiments that we have done with it. Ants have been on this planet, right, for an estimated 150 million years. Let me say that again, 150 million years. Whereas we humans is estimated to be here for about 1.5 million years. Right? Whether we'll be here at 3 million is a question mark. If you put on the news channel and look at it, we will seriously doubt whether we are going to be there, right? Even next 50 years, 100 years, forget about a million. Whereas ant societies, the eusocial societies that I showed you in the previous slide, have been documented to be on this planet for 150 million years. So there must be something that they are doing right in order to be able to survive. Right? So they were there well before the dinosaurs came on the planet, and they continue to be there well after the dinosaurs have gone. Right? But these creatures are still there. So they've got something that is really fascinating and going on. In all this time, ants are diversified a great deal, and today in the world, there are about 13,000 species of ants. We're not talking of individuals or colonies. 13,000 species of ants, right? So enormous diversity. They have been able to get into many different positions in the food web. They are primary herbivores, they are scavengers, they are predators, they are herders. Right? All across the trophic levels, you will find different species of ants. Right? So it's very important to understand what is the organization of this society and how they do things. Further, they engineer our ecosystems. So what do I mean by ecosystem engineers? We all know that soil, when you want to use it for agriculture, has to be ventilated, right? There has to be aeration in the soil. And who is credited with doing most of the aeration? Earthworms. Correct? But the fact is, ants do as much work as the earthworms in aerating the soil. So if we did not have ants in our agricultural ecosystem, the amount of crop you can grow will drastically reduce. Ants are one of the primary predators of other caterpillars, which is what will eat a lot of the pests that will infest our agricultural systems. Not just agricultural system, but even in any natural forest, there are moths, caterpillars, which will eat the vegetation that is present there. And those caterpillars are what are predated by the ants. And hence, those caterpillar numbers will reduce, and it is a very much a part of this integrated system. So, ants engineer our ecosystems such that it is sustainable, and they occupy multiple trophic levels. There are 13,000 species of them present throughout the world. India has a large diversity of these ants, but very little is known about these creatures. So, these are some of the reasons for which I picked it up. But is this enough to only have 13,000 species? We need to see how important are these species? How common does this species present to us? So, for that, we take a measure which is called the biomass. I'm sure you've all heard about biomass. So, if you were to take an ant, desiccate it, and then take its weight, then we call that as the biomass of an organism. So, in the entire globe, if you want to do these exercises and find out what is the biomass of different organisms, you will find that terrestrial arthropods, all insects put together, make a large fragment of this biomass, right? Up to 200 metric tons, but let us say some units of 200 units. In that 200 units, ants constitute about 12 metric tons, right? So it is a substantial chunk of the terrestrial arthropods that is coming from the ants, the whole family of ants. Now, this number by itself does not make too much impact, but what does make an impact is if you compare it with something else. So, if you don't think of all the wild mammals put together, you certainly understand what I mean by wild mammals, right? Lions, tigers, giraffes, chimps, monkeys, everything, all of them put together. All the wild mammals put together, right? How much is their biomass? The number that I have put up over there, wild mammals, is 7. Right? So, 7 is a really smaller part. Ants are easily double that. Right? Close to double of all the wild mammals in the whole world. So, you can see how dominant the ants are in our ecosystem. You may ignore it, we may stamp it, we may put Lakshman Rekha and eradicate them in our houses, right? Nevertheless, they are extremely important in the ecosystems that we live in, right? This is one of the reasons why we do need to look at ants and understand their life in some detail. Now, of all these 13,000 species, there's one particular species that I started working on, and that species is called as Dichoma indica. This, why did I pick this? Because this was just there in my neighborhood. So, in the campus that I live in IISER, Kolkata, I happened to come across this ant, and hence I started working with this particular ant. It was as simple as that. It was not based on research and understanding, okay, what is it that I want to do. I didn't know the name. I had to consult a taxonomist. Then they told me that this is Dichama indica. And then I said, all right, let us look at what is already known about this species. And when I started my work in 2008, 2009, there was a grand total of 1 research paper on Dichama indica. And that research paper was about the genetic variability of this ant as compared to some other ants. That was the only thing that was known about this ant, which lives very much in our backyard. So I said, all right, let us try and understand what is the life of this particular species of ants called Dichama indica. Okay, so now after working for 15 years with 7 PhD students who have finished their PhD and 8 PhD students who are currently doing their PhD and 3 postdocs, I can say we know a very small part of what Dichama indica is. If you want to ask me what put a percentage, how much do you understand this animal? Right? After this 15 years of work, and not just me, I am doing a small part, my students are doing the majority of this work. I can say we have understood 0.01% of this organism. Right? So that is how we would like you to understand that that's what research is about. The more questions you ask and find answers to it, there will be more questions that arises from it. Right? So that is the way research progresses, and we have a small piece of understanding on Dichama indica. So these ants look approximately like what that image shows you there, and each ant is about 1 centimeter long. If any of you are interested, you should ask, is Dichama indicum here in Mysore? Right? The answer is yes, we have seen it, and yes, like sir is telling, certainly there, and I have seen it in the university campus. Today I didn't get a chance to walk around, maybe even in your college it is very much there. But in the university I have certainly seen this species being present. It's about a 1 centimeter black colored ant. It makes small colonies, about 90 on average is its composition, all females, monomorphic, that means they look all identical to each other. All right, and they live in subterranean shallow nests, underground nests which are shallow, and they are known to be present in India, Sri Lanka, Bangladesh, and possibly in Japan as well, like what I'm showing you here in the map. Now, we all know, and if I were to ask you, I drop a piece of sugar here, what happens? Give it half an hour, and you will see a trail of ants that come to this piece of sugar, right? Doesn't matter where, whether it's in your hostel room, whether it's in your bedroom, wherever, right? You put some little sugar, they will come. And they typically make a trail, right? Is there anybody in this room who has not disrupted a trail of ants? Right? This is what we would do. There's a trail of ants, you dip your hand in some water or something, and then you just cut the trail off. Yes? And you will sit and watch for some time. What will they do? Will they stop coming to the sugar altogether? Will they find an alternate route? Right? How do they find this alternate route? So if you are curious, you will sit and watch for some time. And after that, you're like, okay, whatever, you get up and go. Right? But this is an experiment that you have certainly done. At least I certainly did it, and I expect most of you, anybody interested in wildlife, natural history, and any living organism in your vicinity would have done this experiment. So, this species of ant that I work with is incapable of making a trail. This organism cannot make a trail. That is one of the reasons why I picked this organism to study. And I will tell you, instead of a trail, what is it that they do when they have to recruit individuals? All right, so let me proceed. All of our work has been done here in that little spot in our country, West Bengal. And IISER Kolkata is located in West Bengal. And in that, there is a district called Nadia. And on an evening, this is what our campus looks like. This is where our ants are. And we spend hours and hours just sitting with the ants, running with them while they are doing their jobs. Okay, so this is our study area. The vicinity of this IISER-Kolkata campus, basically Nadia district. This is what the animal looks like. If you find something, please take a picture and send it to me. In Kannada, we call black ants which are big as godda, right? This is one kind of godda. And not all goddas are Dichoma indicum, but this is one particular kind of godda. And this one is a solitary forager. That means there will be just one of them walking around. If you see a large number of black ants, which is big, then that is certainly not Dichoma indicum. They are solitary scavengers. So they just walk around and get dead insects and come back to the nest. All right. So for understanding any society, or if I want to understand the behavior of this classroom, for example, the very first thing that you need to do is to have individual identification, right? So all of you know each other for a long time, so you know the names of each and everyone here, right? If you don't know the names, then I'll say, okay, person with yellow shirt or blue dupatta, things like this. So we need some way of identifying each and every member of this group. And if you're even more organized, we'll have a roll number. Correct? We will take attendance, we'll make sure that you're coming to class, keep track of all of this. But individual identification is key. When you look at a trail of ants, almost every one of them look identical. So you don't know what each individual is doing, right? Some may be a bit bigger, some may be smaller. But apart from that, they're all identical. So the very first thing that we have to do when you want to understand how a society works is to provide identification to the members of the society. And the question is, if I want to work with this ant, I need to know how to identify them individually, correct? And the answer for that is this. If the projector will allow you, you can see there are paint marks on the ants, right? So there is a white dash dash. On the other hand, there is another one here, which is white silver silver, right? Then there is a blue dash dash, then there is red white white, and so forth. You can see the red green green, so forth. So as soon as we collect a colony in the wild and bring it to our laboratory, we mark all the females that are in the nest with paint and give them individual identities. And now, after giving such identities, I know what this white dash dash has done as compared to blue dash dash. Are you with me so far? Do you have any questions or something? Clear? So, now I have identification. Now, once I have such identification, I can go on and investigate how the different tasks that these colonies do is performed. Who is doing it? Right? That is what I would like to look in the next stage. And what is the job that I'm interested in? This is a particular unique job, and this job is called colony relocation. Right? What do I mean by relocation? Relocation is a process where you shift your house from one address to a different address. Right? So, some people in this room have relocated their houses? Yes or no? Can you raise your hands if you relocated from one address to another?
We have done not very recently,
but in the past. In the past. So, I don't see all the students. I mean, most of you have been anchored where you were born, mostly, is it? So far, at least. Okay. But for those of you who have conducted this job of relocation, you will realize that this is not a very pleasant job. Agree? If you had a choice, you may not want to do it again. Right? It is extremely tedious. Pack everything, then move it, unpack where to keep, what is broken, all kinds of headache, and everything is heavy, lifting it, keeping it. It's a lot of work. Right? And of course, who is going to do the work? Right? If you can hire somebody and get it done, that is one way of doing it. But if you, in fact, yourself do it, you'll realize that it's an enormous job. Like I told you now, ants also live in nests. Where are these nests? These are under the soil. They are subterranean nests. Now, do you think this society will also have to move from one place to another occasionally? Right? Which is expected. When and under what conditions do you expect it to happen? It will happen when there's flooding. Right? Lot of rain, then of course the water will go into the nest and disrupt the entire nest. Then they have to shift. They will take everything that is with them, go to a different place. Right? So, in all of these 15 years, I've been trying to study how Dichoma indicum moves from one address to another address. There are about 100 individuals. There are about 100 brood items. What is brood? Egg, larva and pupa. And the whole colony as a whole, cohesive group has to go. Because if all other animals go and the queen is left behind, the reproductive individual is left behind, what happens to the colony? The colony will die. Are you with me so far? If the pupa is left behind and you go away, what does that mean? It means the most valuable of your assets, the best developed stage, which is the pupa. You've invested so much in feeding and taking care of this pupa. You leave that behind and go, then you will see that it's a massive loss of investment, right? So you will certainly not leave your jewelry behind when you're going from one house to another. That is the same thing for ants. So the question is, how will these ants find a different house? And if they find the house, how will they transport everything and go there? Huge risk. There are a lot of predators. There are parasites. The nest is inside a dark chamber. Now they have to come out in the sun, get desiccated, correct? And there are predators waiting to eat them up, but they have to go to a different address. So, under this scenario, natural selection will act on them to make this process very, very efficient. So, we want to see what is it that this 150 million year old creatures have come up with as a solution to colony relocation, a task which impacts their survival as well as fitness. Are you with me so far? Do you have any questions? Okay, so I do not have access to a clock or a time thing. So can you tell me at 45 minutes, flag me at 45, so then I can start wrapping up. Yeah, okay. So home is particularly important. Home can be a nest for some organisms like this, which is living in the hollow of a tree, but home is a place that provides you a safe corner to rest and to raise the next generation, correct? It's a place to store resources. It's a place to get away from predators and parasites. So it's very critical in the life of a lot of organisms, including ants. Then the question comes, what is home for Dichoma indicum? Right? I can make them live in a test tube in my laboratory, but that is not their home. So every time you look at an organism, you have to look at them in their natural habitat and understand their point of view. So first we have to discover what is home for Dichoma indicum. Now, this is quite easy to ask, but very difficult to do. How will I find out what does an ant's nest look like? Right? The solution is bring a spade and start digging. Right? Sure, we can dig. And when you dig, what will happen? Large number of ants will come out and go in all different directions. But that will not tell you how the nest is, because the nest would have collapsed when you're digging it. Right? So, the solution to this is to make cast of the nest. What do I mean by making a cast of the nest? As soon as I say cast, what comes to your mind? Fractured hand, you make a cast on your hand. Yes? So the cast will take the shape of your hand. Yes? Similarly, if I can do something to the ant nest, where the material will take the shape of the ant's nest, then I dig it out, then I know the shape and size of the ant nest. Agreed? So, I can put plaster, that is plaster of Paris, into the ant nest. But if any of you have had fractures or if you have worked with plaster of Paris, you will know that it's very brittle. You just have to do this and it will crumble and become bits. So when you go and dig the nest, of course the plaster of Paris will completely fall apart. You will not get any information. So what is the option? We take aluminium and we melt aluminium. Right? It's about 800 degrees in the field where the ants are, and pour aluminium into the nest entrance. Are you with me so far? It's really hot and it is liquid. So it will go through every small nook and corner in the ant nest and it will solidify there. Now when I dig it out and take the soil away, I will have a cast of the ant nest. Now by measuring the cast, I can make out what is the architecture of the ant's nest. Are you with me so far? So the top part there is the entrance as I have labeled it, and I will find the entrance by following animals. So there's a forager who has brought back food and I know she went in here. So this is the entrance. So in that entrance I will pour aluminium. Aluminium will go through every nook and corner and then I recover the nest with this, which is in the bottom panel. Now from this nest I will measure what are the different components of this organism's nest, and then I came to conclusion that colonies live in a single chamber nest. There is only one chamber, relatively simple because ants are known to make very, very complicated nests as well, and we know that across seasons it does not change. What do I mean? I mean that in the monsoon, in the non-monsoon, the summer as well as in the winter, the nests are similar. There is no quantitative change in these nests. There is sophisticated statistics one can do to figure out if there is any differences among nests, and having done that, we know that there is no change across seasons. Okay, now I know what the nest looks like. The next question is, when do colonies have to relocate? Okay, so just because I and you are bored or are interested, we can take a water pipe and go flood the nest. Right? Maybe some of you have done this in the garden. If you don't want an ant nest, you will go dump a lot of water over there thinking that they will get out of here. So that does happen at least partly, and this is part of the natural disturbance, physical disturbance to the nest, which can cause ants to relocate from one address to another. The second reason for which they relocate is lack of resources. There is not enough food in the vicinity. Then also colonies are known to go to a different place. Or if there is too much competition from neighbors, too many of my own species living in close vicinity, then also they are known to relocate. So we wanted to see, when does this happen? This is what literature has to tell us. In the case of Dicama indicum, can we make them move? And if they move, how do they go about doing this task? So while they are moving, the problem is with the young ones. How is it that they will transport the egg, larva and pupa? I told you about honeybees and wasps, and I'm sure you have seen the nests of honeybees and wasps, at least from a distance. Going too close is not a good idea, so from a distance. But you know they make hexagonal cells, and inside that there is egg, larva and pupa, correct? Have you ever seen a wasp taking out that larva and going anywhere? Have you seen honeybees taking out a larva and going anywhere? Or a pupa? No, right? Because they cannot carry it. So what happens when they relocate? Their entire nest is abandoned. All egg, larva, pupa are doomed. They will die. This is a problem, right? It's a huge loss to the colony. The colony's fitness. But unlike them, Ants are known to carry the egg larva pupa with them when they're traveling. Right? This is a huge advantage because they can take their investment from one place to another. Now, there are few organisms which are capable of taking their young ones with them. This gharial is one example. This is a male gharial and it has a territory which is a small pond. And if the water in that pond starts drying, then it will take all the young ones on its back and swim and walk as far as required to give them another place where there is water. These are just a few examples where the young ones are taken with them. There are certain cats which are also capable of transporting their young ones from one den to another by holding them from their neck. But these are only for a little part of their lifestyle and only 2, 3, 4 of their individuals. But now we're talking of hundreds of offspring that is within the nest. So the question is, how do ants solve this problem? And in order to do this, I did a set of experiments within the laboratory. And in today's talk, I want to give you a glimpse of 3 different topics, if I have the time. The cost of moving the young ones. The second is about stealing neighbors. And the last is if individuals or these ants are capable of finding the shortest route or not when they're going from one nest to another. So these are the 3 research topics that I want to touch upon. I'll not go into details. I know that you're not researchers at this time in your career, but I want to give you a glimpse of what is hypothesis testing, how simple experiments are conducted, and how one should analyze the results. All right, so that would be my aim for today. So, like I told you before, this particular species of ant is incapable of making a chemical trail, a pheromone trail, but they have to take the nest mates from one place to another. They have to recruit them. The question is, how do they do it? They do it by this behavior which is called as tandem running. I first just want you to watch what is happening. So is it quite clear that one ant is being followed by another ant along the journey? Can you see that there is physical contact between the 2 ants? The one in front, in this case white dash orange, is being followed by blue dash blue, and throughout the journey, both of them are in physical contact. Right now, the follower is called dash dash red, and she is the queen of the nest. Even she is taken by tandem running by an individual who is in the position number 1 here, and that individual, orange blue dash, is called as the tandem leader. The tandem leader will recruit a single follower at a time, take her from the old nest all the way with physical contact into the new nest, which is another hole in the ground which you see in the center of this picture. Agreed? So, what Dicama indicum does is, the leader, orange, orange, yellow, is being followed by dash, yellow, dash, all along the path in physical contact from the old nest to the new nest. The follower is dropped there and the leader will go back to the old nest, get a second animal and bring her here. Are you with me so far? This process is called tandem recruitment. Okay, it's literally like holding hands and bringing them to a new address. You drop them, go back, get the second person. The automatic next question that comes to your mind is, can some of the followers become leaders? Agreed? If you are awake, then yes, this is the next question that should come to your mind. Will some of the followers who have been taken there in turn become leaders? Or is it just the job of one animal who is going up and down, up and down, up and down? The answer is yes. Some of the followers become leaders, and approximately 15% of the colony become tandem leaders. And they will transport the rest of the 75% into the nest by doing individual tandem runs like this. Okay, including the most important individual, who is the gamergate or the queen. Right? There is a technical reason why we don't call them queen, we call them gamergate, but don't worry about it. The reproductive individual is also tandem run in exactly the same manner. Okay, now, to prove that you are awake, ask me something.
In between something happens, like the heart is broken.
Correct.
There is an accident over there. What is the situation between the
Correct. So here there is no path at all, because in chemical communication, when there is a pheromone on the substrate, then there is a path. Everybody will go in the same path. Here, every leader goes in her own path. So in the leader that you see there, who has discovered the new nest, will take her own path and go. But if there is a physical disturbance in the path that she is taking, like for example, I just put my foot, this vibration is created and that disturbs them, then they will just become completely silent. They will go under the foliage and sit there. Give it 5 minutes or so, then the leader will come out first. And if the coast is all clear, no disturbance, she'll call the follower, and again both will continue back to the new nest. Now, if there's a large-scale disturbance, 45 minutes. If there's a large-scale disturbance, let us say that nest becomes unavailable for some reason. The new nest, I thought it was good, but it may not be. Then what happens? They will stop tandem running. Instead, they will go to a temporary shelter. And from the temporary shelter, they start looking around for other options and go into a different nest. Excuse me. Okay, from the students, we were talking about the reproductive individual or the queen, and she is also tandem run. Anything to ask? Why? Why what? Why is she tandem run? She is another citizen in this colony. She gets no additional privilege. She has to walk the route as like everybody else. Tandem run, say it again. Okay, no, that's a very, very interesting question. So, for all of you who have relocated your houses, when you start looking for another house, you will not find one. You will get 3 or 4 information about 3 or 4 houses. Then you will go visit the house, correct, and check out, oh, this has 2 bedrooms, this has 4 bathrooms, whatever. Right? All of this is done. And then members in the house, somebody will like house A, somebody else will like house B. Correct? Maybe one is closer to my office, the other one, I don't know, I don't like some component of that house, whatever. So I like this. But other members in my house will want a different house. It's entirely possible. But can we shift into 2 houses? The answer is no. So you have to have a consensus. Correct? Similarly, both in honeybees as well as ants, approximately every time these animals look for a new house, the average number of houses that they will find, they will shift into at different degrees, are 5. They will at least find 5 houses. Now the question is, all of the animals have to agree which of the 5 houses to go. Right? This is a process called decision making. Now, one can understand how such decisions are made in such societies. And in the honeybee colonies, what we know is that there is a threshold. All of the animals who are looking for nests are told that there is a nest in this particular location using the waggle dance. Have you heard about the waggle dance? Following the waggle dance, other animals who are doing this job of searching for nests will go to the same location. Now, if there are 3 animals which have gone to that location and they like that place then they will stay in that place. And if there's a threshold of 12 bees in that new nest, then a consensus is reached that this is the best option. I know of another 3 nests, but this one which you have taken me there is okay by my standard. Like that, if 12 bees tell, yes, this is all right, a threshold is reached, then the whole colony will shift into that. Okay, this is known for honeybees.
I have a question. The nest is made new or existing one?
Yes, so many ants, when they're founding their nest, will make a small hole. It's only one queen who starts the nest. She will make a 1 centimeter hole and sit inside there. The workers will later on expand the nest. But this species of ant is a scavenger by nature, and it goes into pre-existing holes and modifies the nest to make it into that single chamber and a runoff tunnel that I have shown. So this will go into pre-existing holes and crevices in the ground and then modify it. Okay. So in ants and in another species of ant called Temnothorax, we know that this threshold effect works. But in Dicamba indicum, we did several experiments to find out if threshold works. And what we know is, if I have a large number of animals there, it does not speeden up the process of deciding on the nest. But if I keep removing everybody who comes to that nest, then it delays the decision for going into that nest. But the decision is with the individual tandem leader. If the tandem leader likes the nest, she will bring her followers to that nest. If she does not like it, she will find an alternate nest and take them. Then tandem leaders from these 2 different places will tandem run each other. And hence, this animal will know of that nest, this other animal will get to know of this, and then they decide which is the best. Right? And the colony will shift into that. So the whole process, if we were to observe in nature, it takes about 6.5 hours. They go to 5 different nests and from there merge into just one single nest. Okay, now the question that I was trying to get you all to ask is, when does the queen do tandem running? Does she go to the new nest in the beginning, or does she go after everybody has been taken there? Yes, what is your guess? When do you think she will go? In the beginning or in the end? There are other options. I have told you only 2. What do you think? In fact, we have done several hundred experiments, and in all of them, the gamma gate will go in the middle of the relocation. Why middle? The risks are optimal. Being here is as risky as going there. That is the time when the gamma gate goes from one place to another. Okay, so you have an idea of tandem running. Now the question we were starting off with is, what happens to the brood? Oops, I thought this was charging. Lose connection here. Is there any other plug point that I can put into? No, it's telling me low battery. So it should be red here, but it's showing me white. No, it's still showing as white. It's coming, it must be working now. This light came on, so it should work. Yeah.
Charging this one. This is charging.
Well, it's the same thing. We can put this there so that the talk is not interrupted. Alva.
Sure.
I will know. Oh, so this is mine is charging there, is it? Okay. Okay, it looks like my laptop needs to restart from beginning. So in the interim, we can anyway continue. So we were looking at how if they will take all the egg larva pupa, it's stupid not to take it because why? You will lose fitness. So will they carry it or not? Now, ants are known to carry large, be capable of carrying large weight with them. Right? You would have seen several ants carrying. They can carry a whole ant. Right? They can carry large particles of food. They can carry large prey, but together. Right? 10, 15 ants together is taking one big cockroach and going. Right? So we see this all the time. Ants are capable of carrying large loads. So we will not be surprised if they will carry their young ones. Okay. Now, before I tell you the answer, what would be a good way of carrying it? One way is the tandem leader can first go take one adult animal, drop her, come back, then take one pupa, go drop it, come back. Yes, it is entirely possible to do. Is there any other way of doing it? Something more efficient. Exactly. So, if the tandem leader is holding one pupa in its mandibles and also has a follower, even better, if the follower has a pupa and the leader has a pupa, so at the same time, you are transporting 4 items, one adult and 2 brood items. Correct? So, we wanted to see what is it that our ants would do. And to know the answer, we will have to wait for my machine. Okay. Okay. So, what we found out is that in this species of ants, we have a coupled adult brood transport, and almost all the cases, the follower holds a brood item in its mandibles, in its mouth. So like I have made a drawing there, the follower has a pupa and the leader is leading that follower with the pupa. So at the same time, leader is taking 2 items from the old nest to the new nest. Okay, so they perform coupled brood transport, and it seems that this is relatively rare in the ant societies. Whatever literature we have so far is where there is a chemical trail, and along that trail, what you find is animals who are carrying the brood by themselves and walking on this trail. Alright, so now, what does it mean to do an experiment with ants? Can I go today, it's a sunny day, looks good, not very hot. So shall I sit down and look at some ants and say, okay, this is my answer? That is not the way it is done. You need to have a hypothesis, and then you need to say, I'm going to perform this experiment either in the natural habitat or the controlled conditions within my laboratory to come up with an answer for this. Right? And in many cases, we do manipulative experiments, which will let me clearly understand the connection between the cause and the consequence. All right. So, for coming to this conclusion that coupled brood transport happens, we followed 12 different unique colonies and 1,205 transports by all the ants in these colonies in the natural habitat, and looked at 141 transporters, unique transporters. And only then, after analyzing all this data, we come to the conclusion that approximately 85% of the colony's brood is transported by this coupled adult transport. Alright, so it's not like one evening I went for a walk, I saw one ant going with one pupa and I say, okay, this is it. It's not at all like that. Now, in the next step, if they're capable of brood transport, then can they do anything else with it? How efficient is this process? If I were to ask you to go from Yuvarajas to Maharani's college by walking, you will take X amount of time. If I give you one suitcase, and the suitcase is your own weight, the same as your weight, how long will you take? Right? If we can manage at all in a reasonable amount of time and we don't collapse on the way, we'll take easily double, triple amount of time. Now you're thinking of a lovely road. Right? Ants don't have the road. You saw where they're going. There are grasses, there are stones, there is water. You have to go through all of that. So it's not easy to collect 100 adults and 100 brood items and go from one place to another, including all the stored resources. What is stored resource for this ant? It is items of food and their favorite food item, which is termites. So if that is in the nest, they will take that also. Alright, so we ask the question, if they're doing it for 100 million years, how well have they optimized this problem? How do you do this question? A BSMS student who was doing his master's thesis with me, and I performed this experiment. You do a simple process where you bring a colony to the lab, take away all the brood. Agreed? No suitcases. Then measure how fast the colony will go. Same distance. How fast will they go? Is that enough? No. Give them double suitcases. 100 brood items were already there. Now I'll give them another 100. And then now compare what is the problem? How much longer will they take? If they are very well optimized with this problem, then they should not take significantly longer. Correct? Within the same amount of time, they should be able to manage. The question is, what did the colonies do? Are you with me? This is a manipulative experiment. It looks at the hypothesis about regarding the cost of transportation of brood in the nest. So, as mentioned here, there is a control colony where there is no manipulation and they are going the exact same distance from place A to place B. Same area. Okay. In that same area, in the same arena, we give B minus, that means colonies without brood, and see how long they take. And we give them B plus, double the amount of brood that they had, and now see how long they take. Alright, so what do I find? I first of all find that when I give them additional brood, many colonies decided not to relocate at all. They just said, no, this is not possible for us to go there. I'll take the risk of being in a destroyed nest. Right? So that is the B plus colony bar. Only 7 colonies out of 12 moved. Alright? But when there is no brood or control colonies, all the cases they moved. But for the colonies that did move, I don't know how much statistics you know, and I'm not going to bore you with the details, but this is called as a box and whisker plot. And in this, if I had a pointer, it would be useful, but doesn't matter. Just think of one of those rectangular boxes and a line in the middle. The line in the middle is the median of the data, the midpoint of the data. And all the dots are the actual data from the experiments that you do. You should see that they discover around the same time, but they take longer to decide to relocate, okay, which is the latency bar when there is extra brood. But if they decide to relocate, they take the same amount of time as the control colonies, even though their brood is double. All right, and if there is no brood, does the process become double fast? The answer is no, it takes the same time. Right? So they have optimized this process of carrying the luggage of their future fitness with them. So having no brood does not enhance the relocation dynamics, and having extra brood significantly reduces the chances of relocation, but they do so without any extra time being invested. And remember, time is extremely important in these processes, because the more time they're out and exposed, the more vulnerable they are. Right? Vulnerable to parasites and predators. Now, I don't know how much more time I have, but I'll touch upon one more item. 1 hour is done. Okay, but there was a little glitch with your computer. So I will take another about 5-7 minutes extra. Now, you think predators and others are the problem, parasites. There is one other problem. This problem is something that when I tell you, you will say, oh, of course. But otherwise, it will not occur to you. I was telling you to go with suitcases from here to different places. You can go to the railway station with your suitcases, multiple suitcases. And what will you be worried about? Say it again. Safety. Somebody can steal it. Right? Absolutely. So, exactly like any valuable item, there are thieves waiting to steal valuable items. In this case, it is the brood of the colony. Right? So, I want to give you just an introduction to what stealing laborers do. Who is stealing? Other human beings, correct in our case? Similarly, other dicama indicum, conspecifics, steal from each other. Okay? Now, you would most probably not believe me. You'll say, Arey, what is this? It's just a story. Why will an ant steal from another ant? So, always, as an experimental person, you need proof. And the proof is to see an act of stealing. Right? And to catch a person who is stealing, you need a camera. That's quite good enough. But you need something else, which is identification. Right? So what we did, we got one colony, marked them all with one kind of paint. Then we got another colony, marked it with a different color. And then we see red color ants go into yellow color ant nest and steal things from there. Alright, so let me show, if you're interested, a video of a thief. Tell me if you can identify who the thief is. This is the colony. There is a lot of pupa all marked, and they're in the middle of the nest, in the corner of the nest. Can you see? Shall I play it again? Yes. So, this time I'll tell you, I can't point out to you. So now an animal is coming in. She is blue, green. She sneaks in, takes the pupa, out she goes. You saw this? Shall I play it again? She has not yet come in. There she is, blue, green, sneaking into the nest, picks the pupa, and goes away. Yes? So, of course, one has to define what I mean by stealing. Okay? And the definition of stealing is to take another's property without permission and without intention to return. Right? So, I'm sure in this case they don't mean to return. And we wanted to understand this phenomena in greater detail, and hence we did a lot of experiments regarding stealing laborers, and we saw after doing about 15 replicates, around 15 items were stolen. Okay? And in these 15 items, 14 were pupa. Why only pupa? Pupa is the most invested item. Pupa is a cocoon, right? From that, an adult ant is ready to emerge. There is no need of any investment, no need to take care of this pupa. You are going to get an adult immediately. Right? Now, once this is taken back to the nest, will the ants eat it up? It's a source of food, right? They can as well eat it up. So we followed what happens to them in the nest, and we found out it is not for eating. It is for eclosion of the adult. And what do? That eclosed animal becomes a slave inside the host nest. That slave will do all the work that is required for the nest. Are you with me? So it is to make slaves that they're stealing. Alright. So, does everybody become thieves? The answer is no. Less than 2% of a colony become thieves. Alright? But once an animal becomes a thief, she will go and attempt again and again and again. The record that we have seen is 36 attempts by one particular ant to steal. And in that so many attempts, on average, 75% of the times they are successful. And they always bring pupa back to the nest. Okay, so stealing laborers are a problem. Then we went on to see what are the characters of these thieves. And we find that the individual who is exiting from the nest, if I were to mark her speed, not when she's going into the nest, but when she's taking the stolen item and going away, her speed is faster than what a cheetah does. Right? So among mammals, cheetah is known to be the fastest organism. Now, can I just say how many kilometers speed? Of course not. Of course, the cheetah is a much bigger animal, correct? So how do? You have to look at the body size. How many body size per unit time the cheetah goes, and look at how much Dacama is and how fast she goes. And it turns out that Dacama is faster, but only in that stretch after she has taken the pupa to bring it back to her nest. Rest of the time they are nowhere close to the cheetah. But this, they are really, really good. So, we did this large set of experiments in order to come there, but in today I will not get into the direct and indirect observations. We discovered rather that there is intraspecific stealing, and we were the first ones to report it for all the tropics. Only in temperate areas ants were known to steal from each other, but in the tropics they said, no, there is no problem here, the temperature is very good, individuals will not steal. But that hypothesis is completely wrong. We were the first to report in the world that in temperate regions also such things happen in the society. So, we discovered intraspecific stealing in tropical regions and find that it acts as a major cost for relocation. About 30% of the nest's pupa is stolen. That is not a small number. 30% of all your investment is stolen every time you relocate. Please say it again.
If they don't work, what to pay?
If they don't work, the slaves, okay, the slaves, the term is very fancy, but the point is the slave is born in that nest, enclosed in that nest, and she does not know that it is not her nest. She thinks that that is her nest, and she will work like every other animal in that nest.
It's like taking a helping hand.
Yes, we call them slave because it is not a willing helping hand. And second, there is no relatedness between this slave individual and the queen. But in the normal nest, all animals are the daughters of the queen or sisters of the queen. So there's a very high genetic relatedness. But there you are a stranger, but you are made to work, and hence we call them as slaves.
When there is no genetic relationship, then there is no genetic conflict. Behavioral conflict, generally there should be there. Behavioral conflict.
So it depends on what is the number. And if all of those slaves are coming from one particular colony, so if they can make a cluster, then there's a chance of this. But if there are two animals, and if these two animals are from two different colonies, and they don't even know that this is not my nest. Because wherever the animal is eclosing, they think that that is their nest. So they're unaware of this. But with evolutionary time, if there were a large number of slaves from one species, one colony, then yes, we can expect certain differences. But to date, we have not seen such differences. It is unlikely in the colonies of hundreds, if there are tens of thousands of workers, and if the raids are from just one or two colonies, this can emerge.
My question is, in these colonies, movement of these workers is never represented. The gamma rays usually theoretically specific precision period. It is quite different but introduced one.
Okay, yes. So that's an important point, as in the world of these insects, their input is through a lot of olfaction, through chemicals that are present in the nest. So imagine that you lived in a house for 75% of your time where it is completely dark. Right? Then how will you navigate? How will you do your functions? By the processes of touching and smelling. Right? For example. So the ant's world is not a visual world as much as it is an olfactory world. All right. So the queen, to say that she is present, will produce chemicals which are called pheromones, and by smelling that pheromones, others in the nest know that the queen is there and she is healthy. All right. So what sir is asking is, this naive animal does not have any chemical of the nest. How does that manage? This is because every newly eclosing ant is a blank slate. They don't come with very minimal chemicals on their body, and those chemicals are to just signify which species they belong to, not which nest they belong to. But within 5 days of their lifetime, their cuticle will accumulate the pheromones that are in its environment. And some small part is produced by themselves as well. And then this becomes their character. Okay. So, if this animal ecloses in a new nest, and if she's there for 5 days, she will acquire all the smell of that nest. And nobody will know that she is not related to them. Right? Everybody is homogenized. This is what happens. After 5 days, if I take an ant from this colony and put her here, she will be killed. But within that 5 days, there is no problem. There are blank slate, chemicals will get deposited on it, and they get accepted into the colony. Are you with me so far? Now, another thing this brings me to is, even on a trail, I've been asked this question so many times, that there are ants that are going in 2 different directions, and they'll touch each other, then move forward. We've always seen this, correct? What are they doing? That is exactly what the question that sir was asking. With their antenna, they touch the body of the other ant and see whether this is from my colony or not. If she is from my colony, they will just touch each other, go in their direction. If it is not of the same colony, then they will touch a little more, produce pheromones which are called alarm pheromones. They will themselves start fighting, and because of the alarm pheromone, other ants will come and join the fight. In this trail, it belongs to a particular colony. Another colony comes here, it's gone. All right, are you with me so far? Okay, so now let me quickly wrap up. Yes, please. About the?
It has stolen. In what way it will help the thief is stolen? Because when I see this one, this should be helpful for me. I will trade it or I will keep it, I will trade it.
Correct, correct. So what is the profit for the thief? Correct. Correct.
How it will track, it has to follow me or it has to be safe for him?
Correct. So what is the profit for the thief? Right, that's what you're asking me. Correct. So yeah, what is the benefit for the thief? Now, first of all, we should understand there's higher risk for the thief. Yeah. Because it is going into the other nest. Other ants can catch it and tear it apart, make it into bits. Right? So that risk is there. Now, what is the benefit? The whole colony is getting a benefit because they're getting slaves. The workforce will increase. Correct. But what is the benefit for the thief? Right? The answer is, I don't know. We do not know.
Because there is a law out of this is
Yes, yes.
there is no benefit.
Correct.
Why do we
Correct.
indulge?
Exactly.
That is the
Exactly.
of human being.
Yes, yes. So only 2%, absolutely. So, oh, correct. So, there is a benefit to the colony. However, to the individual, is there any benefit? We do not know. Now, why I'm saying this is, only 2% of the colony become thieves. And once they become thieves, however, they do as many trips as possible. And they go try, come back, try, come back. Well, as long as there is a source available, they'll try to steal. So, the 2%, yes, the 2% will go on attempting again and again.
They had it
Possibly.
Who people, me or
Yes. Yes, we can say that they're specialized in stealing. So again, to just think of it, 150 million years, right? These ants have been there, and they're using some very similar strategies of thieves that we humans do too. Right? It's an ant 1 centimeter long, less than 1 millimeter brain size. Correct? But it's a phenomena that is ambiguous. It happens there, it happens here, it happens pretty much in the same way. What is the benefit for the thief? If we could communicate with ants and ask them, possibly we'll have some answers. Right? But as of now, all I can say is that the ant is doing it for the benefit of the colony. Right? There is benefits for the colony. Now, most of the jobs that ants are doing is for the benefit of the colony. All the food that it collects, it cannot eat because it does not have the digestive capacity to swallow big chunks of food in its digestive system. It will collect the colony, give it to the larva. The larva will digest it and it will give it a fluid. All right? So, whether it is bringing food, whether it's bringing resources, whether it is defending, there is not any great benefit for the individual, but it is done for the colony as a whole. So, stealing.
Goodwill of the friends. I steal my job is to steal. I visit my friends. Whoever wants it. So that I get good. Yeah, here is a map. We teach for the teaching for them.
Yes. Possibly they may have prestige, who knows, right? The point is, we cannot communicate with these organisms. So we can only observe and then conclude as to what is it that they're doing. So if another alien species was observing this classroom, right, then they will say, yeah, you were all just sitting around. They will never know that we're talking about something else like here, right? So for that, you need to understand their language. And we, as scientists, are very far away from understanding the language of any other organism, including ants. Right? Even chimps, we cannot understand their language. To the extent that even among humans ourselves, we are not very good at communicating and we have a lot of misunderstanding. Correct? So, until we can get into the Umwelt, so the technical term for this is Umwelt, we will not be able to understand what is going on in the minds of these organisms. What do they communicate? Is there prestige at all in these societies? Is there only just work for the society or is there anything for the individual? We will never know this until we understand the means by which they communicate with each other. Okay, so given the time and how I was very pleased with all the interactions, I wanted to tell you whether they take the shortest path, but I will not do that. Instead, go to the summary and tell you that today we spoke about the cost of moving the young ones, the egg, larva, pupa, and we find that relocation is optimized to integrate moving the brood in this species of ants. There are stealing neighbors. Pupa is the one that is lost most of the time, and it's a huge issue even in the tropics. The other experiments would have told you that tandem breeders are very efficient. They show about 80% efficiency in finding the shortest path from the old nest to the new nest, even though they encounter it for the first time. And remember, there is no chemical trail. So the ant's neural system is processing information along that path that you saw to make an 80% efficient trail at individual level, right? Which is absolutely remarkable. And maybe some other time I can tell you how we discovered this. So overall, we are using this paradigm of moving from old nest to a new nest. And using that paradigm, we look at several different questions. I touched a little bit upon navigation. We look at traffic rules. So if there are multiple ants going up and down, there can be congestion. Right? How do ants solve this problem of traffic along the routes? We look a little bit at the neurobiology and the memory of ants. How do tandem leaders remember which nest is there, where it is, how to get over there? How was it? What are the dimensions that they remember? So things like that we look at. We look at problem solving in ants. If we give them a problem, can they solve it? And of course, the problem has to be relevant to their life history. How do they make decisions? There is nest A, B, C available, and there are trade-offs. One nest is a bad quality, but it is near my old nest. Whereas another nest is excellent quality, but it's very far away. So how do they deal with these kinds of trade-offs? Then we can look at other tasks that the ants are doing, and of course other insects, and how they solve these similar problems. So basically the conclusion and the take-home message is understanding proximate mechanisms by which organisms overcome the challenges they face is very, very essential. And only this will allow us to appreciate the lives of these organisms and how evolutionary history has fashioned these solutions that these societies have come up with. Now, more specifically, being able to efficiently relocate gives ants a selective advantage over evolutionary time. Unlike other social insects like wasps and bees, these can take all their investments and go to a different place, and this is a huge advantage in their natural habitats. And if organisms have to colonize new areas, then you can imagine that it would be a great idea to take all your young ones and go there instead of going there and starting from scratch. Right? So these are the two things that I want you to keep in mind. And I should tell you that all the names of my PhD students, and Purmayan is the master student that I was talking to you about, who have done the work I have had the privilege to address today, is mentioned here. IISER Kolkata, my host institute, and SERB, that is the Science and Research Board, which gives me grant money in order to do the experiments that I function. So without that, I will not be able to function. We need very little money to do it, and a lot of patience and a fantastic mind in order to find the answers that we do. And all of that brilliant work is done by my students who are mentioned here. Thank you very much for the opportunity and all your patience. Yes, yes, you're most welcome.
Lifespan.
Say it again, please.
Lifespan. What is the lifespan?
What is the lifespan? So,
And the number of times relocation.
correct. So, in the lab, the adults will live, the workers will live about 3-4 months. But the gamergate or the reproductive has lived for about 2 years. So always the queen's lifetime is much longer than a worker lifetime. That is true for other social insect colonies as well. Now, in the natural habitat, to find out this lifetime is very, very difficult. Because I'll have to go expose them, mark them, and that itself may kill them. Yes. So I will not be able to tell in the natural habitat what it happens. But we know for sure that the reproductive lives at least 2 times as long as the workers do. Now, the next question is, how often do they relocate? So this is also something very, very difficult to find out. Why? Because we go to the natural habitat and we have to mark every nest that is available and keep on following what is to do. So we have done it for a year now. And every alternate day, 3 of my students will go to the same place, look whether the ants are staying in that same hole or not. Or if they've moved, where have they moved? So we have to keep track of that. So we took a small bit of land, about 250 square feet. And in that, we found over this 1 year time scale, about 70 nests. And we tracked them. But we're in the process of analyzing to see how often do they move. it will be also based on seasons. So in some seasons they would go faster, whereas some other seasons they will not go. And of course, there's a lot of anthropomorphic disturbances. So what human activities we do will influence what the ants are doing because they live in such close vicinity. So we have found a piece of land where there is relatively less anthropomorphic activity, but not entirely devoid of it. So the answer to the second part is we're trying to work it out with a small sample size, but across 1 year of work. Hopefully in the near future we'll have an answer.
Why two different species cannot live?
Why she's asking why two different species cannot live together. Even of the same species, if the offspring is from one gamergate and another gamergate, they do not come together and live. They will only one gamergate's offspring will be inside a nest. Now, some species of ants can have multiple queens in them, but then there are millions of workers within the same nest. In this species, even two gamergates will not live together at the same time. Now, if you move on to a totally different scenario, in the temperate regions, there are slave makers of species A who will go and raid species B, and their entire workforce is species B. So in that case, two species are living together, but it is only to the benefit of A, because only A will reproduce. But otherwise, two species and the offspring from two different colonies also will not merge and live together. There is a lot of genetic reasons for it, and there is a lot of relatedness issues which we think supports this idea, and it was initially proposed in 1930s by Hamilton. There is ample proof that that is the case. It's to do with relatedness. All the members within a colony are very closely related to each other.
Yes, that has two very important aspects that have been involved in evolution. One is non-existence of polymorphism. Second one is loss of wild movement.
Loss of?
Wild movement, right.
Okay.
Wild movement is completely lost
Correct.
here.
Correct.
So I think this loss are not a gain.
Okay. See, the trail, the easier question to answer is the trail. Now, trail is required when you have to recruit organisms. Correct. So, if I have found a huge mango and one ant can only bring so much of a mango back to the nest, right? I need hundreds of ants in order to come to that mango, take the food back efficiently. All of Dicama are scavengers. What do? If they find a big dead cockroach, they will ignore it. They don't have the mandibles to cut it into bits and bring it back. Okay, they will ignore it. But instead, if they find one dead ant, they will bring it back. Do they ever recruit to food? In Dicama indicum, we have done experiments to see whether they will recruit. We gave them high protein source, high sugar source, and saw whether will they recruit in any circumstances. The answer is they do not recruit. So my simple-minded answer to this is that in Dicama, there was no need for a trail pheromone. Only when they're relocating, they need to recruit each other. But on a normal, yes, yes. So on a normal basis, they do not recruit for food. So there is no need for it. And please remember, for everything that you make a trail pheromone, there is a cost associated. Several glands have to change, several chemicals which are very expensive have to be made, and they have to be kept in reserve. Right? That's a cost. Unless there's a reason for that cost to exist, we will not expect evolution to just have this anyway in the abdomen, whether you use it or not. So, in this particular case, my personal idea is that there is no need for a trail pheromone in Dicama, and only during relocation they recruit. Otherwise, they do not recruit. Hence, the cost is too high in order to get this. The second question that Sir was asking is about the reproduction in Dicama. It's a very, very interesting story. Like I told you, all the ants in this nest are identical. They're monomorphic. So you cannot tell one apart from the other. Even if you see in a microscope also, you cannot make out. Now, what is special about the gamergate is that in my diagram also I have shown you okay, I just needed the pointer. I have drawn something over here on the thorax of the ant. This is not to scale, it's just a drawing that I made, but this structure is called as the jamma. Right? So in dicama, it turns out that all animals that eclose from the pupa are eclosing with a pair of thoracic glands called jamma. The resident gamergate will go to this newly eclosing animal and mutilate the jamma. What does it mean? Mutilate means cut it off and make her into a worker. Only if the jamma remains on the body, then the animal will do something called a sexual calling, find a male, mate with that male, and become the future gamergate of the nest. Are you with me so far? Now, this is in contrast to other social insects, other ants, where queens are eclosing with wings. So if you see after a big rain shower, there's large number of ant queens and males which will come to different light sources. These are the future reproductives. They're looking for mates and they have wings. But in dicama, nobody will have wings except the males. Males have wings, but none of the females will have wings. Instead, they have this jamma on their body, and as an adult, on being mutilated, they become workers. Now, this is a very, very interesting phenomena. It is well worth studying, and we have started some bit of experiments on mutilation, but I do not right now have the resources to keep my colonies for long periods of time, because to study mutilation, I need them to be in my lab for about a year, 2 years, and be healthy. Yeah, it is. Right now, it is not the case. It used to be, and it was a different species, not Dicama indica, which was called Dicama cellulens. Yes. Right now, it is not. And in my lab, I would like to have it there to be able to work with it, and the organism is right there. So, I have not had the chance to work so much on the reproductive component. Hopefully, with time, I'll be able to do it. They don't survive in my lab for long enough. Yeah. Thank you. Thank you.
Regarding to reproduction, to produce such an enormous number of ants, and queen is one or something.
Yes.
In that case, is there any asexual type of reproduction such as parthenogenesis?
Okay. So in the ant system, the current record is held by an ant. This colony is in the US right now. The female ants have a specialized organ called the spermatheca, in which after mating they will store the sperm. So what is known is, in their mating flight, they may mate with multiple males, store all the sperm in the spermatheca, and come back into the nest. And in her whole lifetime, she will not go out of the nest. She will be just in the nest. The current the record is 22 years. One queen who mated 22 years back is even now laying viable females and male eggs from the sperm that she collected 22 years back. Now, the second component that I have to mention is the spermatheca, the gland, has a opening which can be adjusted. It can be opened or closed. So, what the female queen does is, if she keeps the spermatheca closed and then lays an egg, it is unfertilized egg. Correct? There is no fertilization because the sperm did not come in contact. Then this haploid egg develops into a male ant. Okay. If it is fertilized, then it becomes a female. So, not parthenogenesis, so to speak, but there is a method by which a single copy of all the chromosomes will give rise to mates. And the queen has control of the spermatheca. So she can regulate whether she wants female daughters or she wants sons.
I think that kind of process is random, not necessarily based on special type of how to choose. It is random type.
No. The queen has control of the spermatheca.
It is optional.
It is optional, and how do we know this? By dissections, you know that the valve can be open closed, but in addition, Males are produced seasonally. So if it was chance, then sometimes you get male, sometimes you'll get female. But that is not the case. There is a clear calendar of when to produce male, when to produce female. And this is followed by every member of the species. Yes. So it is, yes. So it is regulated. It is regulated. Okay. Any other questions? Or
We are
should
telling about the trade.
correct?
Is it because the size of the population or the members are
Yes.
very less when compared to the other trade, that trade is there?
Yes, yes, absolutely. So when I'm talking about the costs, in order to recruit 100 individuals, the cost of preparing that chemical and storing it is too large for the species. But if there are 10,000 animals that I have to recruit, then it makes sense to produce this thing. And the Dicama indicum is by no means a representative ant species. This is at the lower end of the ant spectrum. There are colonies where there are about a dozen individuals also. So this is not that small, but hundreds is very small. Tens of thousands would be the average number of animals in an ant nest. But I do not work with this kind of things because to follow individually mark 10,000 ants and keep them and follow what they're doing is a totally different level of complication, which is not what fascinates me. This 100 is what fascinates me, and this is a number that I can grapple with. I can keep usefully in my laboratory and be following what each individual is doing.
Breed in the laboratory.
No, so yeah, that is a problem. The ants are available in plenty in my vicinity. That is my biggest strength. So whenever I get the colony to my lab, they survive for about 15 days. And why do they not survive? I told you that I've been a part of IISER systems from its initiation almost. I joined in 2008. We have ourselves relocated our laboratory 4 times. And in each case, we have done the electric work and constructed the building and planned it and all of this. So what happens is that there's a lot of physical disturbance. And it's a new system. We put a centralized AC. But that does not mean that the temperatures will be as well regulated as what we want. In addition, my neighboring lab is an immunology lab. They want it at 16 degrees, I want it at 26 degrees. And of course, the air condition is very confused as to what is to be happening. And as a consequence, my ants don't live long. So we are building a biome where we can house not just ants, but other kinds of organisms which have specialized requirements. Hopefully in 2 years, when I relocate for the 5th time, I can have my colonies live for much longer. But now that is my limiting factor. Whatever I have to do, I have to do it 15 days and release the colony back into nature. And anything I want to do, get a fresh colony. So that is the negative side of it. Yeah. Yes, so this is an enamel paint. So that is the only imported item in my lab, so to speak. It's a particular company called Testers. It is manufactured in the USA and it is used for kids to paint on toys. And why do we use this? Because it has been tested on insects and we know that it is non-toxic. See, otherwise I can just get whatever that is available, Asian paints I can start using. But we don't know what is the consequence of this on the ant. Right? It may die, it may, and like I told you, their world is very much chemical. And on top of that, you put Asian paint, you don't know what you're doing. And we want to see animals in their natural habitat doing their natural things, right? Not in the very artificial system. So we have to be careful of what we do. And this is very quick drying. Otherwise, the other problem is, ants groom themselves, right? They keep on cleaning themselves. So the moment you put paint, they'll do this and that's it. Antenna will get stuck and then the ant will die more or less. Yes, yes. So that is very critical and we need some skill. Every ant is held by hand, a very small amount of paint is put. So that requires a bit of training.
Without harming them.
Without harming, exactly. Because yes, and if you harm, then okay, release the colony, go get another one and come. So it's a lot more work. Yeah, sure.
I hope you all had a nice time. And it was already 4:50. Yeah, so yeah, on behalf of Cosmos, I think you might have heard it. In Mysore has been started in Varda, and we are doing education related. So today we had an opportunity to host Professor Sumana here at the on behalf of Cosmos, I would like to thank you, Yuvaraja College, University of Mysore, the principal, all the academic and non-teaching staff, and all the faculty, especially and all of you for coming here. And I thank everyone who made this talk possible. And thanks to you also. Thank you. If you have any more questions, you can talk to her after. We are to, I think we are ready to talk. And thank you.
You did good.